I'd like to thank the organizers of this summit for inviting me to present our work on pyruvate dehydrogenase deficiency today. And I'll be focusing on our experience with piloting newborn screening for this disorder and the prospects of novel target-based small molecule therapeutics for this disorder. I have no conflicts of interest to report. My outline is as follows. I'll be talking to you about PDC deficiency as it relates to enzymology, some of its clinical features, the molecular genetics, the E1 structure, which is a component of the PDC complex, and the therapeutic options that are out there right now. And that would be a nice segue to introducing our approach to the pilot newborn screening protocol and its limitation and the utility of some of the biomarkers we have used for identifying early patients with PDC deficiency. And then finally, I'll be talking about our work with PDHA1 specific target-based small molecule therapies. And in order to implement that, the importance of knowing how specific missense mutations are perturbing the E1 structure and function, and to be able to then identify and verify E1 targets for smaller molecules that hopefully will bind and restore PDC activity and have a benefit to the patients. So PDC deficiency in general is a mitochondrial neurometabolic disorder of carbohydrate glucose oxidation that mostly affects the brain and leads to decreased ATP production and energy deficit. So it's a mitochondrial energy deficit disorder. It's a major cause of primary congenital lactic acidosis. It's the second most common genetically resolved mitochondrial disorder in the North American Mitochondrial Disorders Consortium Registry, second only to Paul G related mitochondrial disorders. And from our work in piloting for newborn screening, we know that it's at least 1 in 40,000 live births annually will be affected with PDC deficiency in North America. This is from Ohio data, where the birth rate is about 130,000 or so newborns born annually. That translates to about 90 newborns annually in the United States will present themselves with PDC deficiency at the newborn. These incidents is in the range of what we know of GA1 in Ohio, 1 in 60,000, and VLCAD, a very long chain fatty acid oxidation disorder, 1 in 40,000 in Ohio. PDC is a complex that sort of links the end product of glycolysis, which is pyruvate, to the start of the TCA cycle at 12 o'clock in the circle here. And so it takes a 3-carbon keto acid and decarboxylates this into a 2-carbon CoA. In the process, generates NADH, and to feed into TCA cycle. TCA cycle generates reducing potential that then goes into the electron transfer chain to generate ATP. I want to bring to your attention, around 6 o'clock here, alpha-ketoglutarate dehydrogenase complex is another dehydrogenase that takes alpha-ketoglutarate and makes succinyl-CoA. And these are irreversible steps. What happens when there's a functional enzymatic deficit of PDC? Essentially, pyruvate levels go up. Pyruvate can get transaminated to generate alanine, which goes up. Pyruvate then gets reduced to lactate by lactate dehydrogenase, and lactate goes up. At levels greater than 3 millimolar, lactate inactivates the first enzyme in the proline catabolism pathway, which is proline oxidase. And as a consequence, proline goes up. What is unique with PDC deficiency is lactate to pyruvate ratio stays normal. So this ratio of lactate and pyruvate, the ratio stays normal, as opposed to primary mitochondrial disorders where pyruvate is not increased, but lactate is increased because of the shift in the redox potential in NADH and NAD ratios. And so that lactate to pyruvate ratio can be as high as 30, 40, 50 or more. The clinical features of PDC deficiency is broad, with neonatal onset, infantile form, and late onset in children and adults, where there's minimal disease burden, namely ataxia or atypical Guillain-Barré sort of picture with longevity and minimal, if any, cognitive delay. But for the most part, the typical patients will have hypotonia, 89% of them, developmental delay, 83% of them, ventriculomegaly up to 85% of them, corpus callosum abnormalities, about 55% of them, seizures, about 57% of them at the most, Lee syndrome, about a third, and ataxia, about 25%. Of key is the median age of diagnosis is 12 months. And the mean is 31 months. So it's quite delayed in when we are diagnosing patients with PDC deficiency. Females have better survival, but surviving females are more severely affected. And in general, for those typical cases, most patients die, 90% of them within 4 years of age. I'm presenting 3 cases to sort of highlight similarities or differences. Case number 1 is a female, 13-month-old, generalized hypotonia, developmental delay with some paucity of white matter and a thin corpus callosum and patchy enhancement of caudate and lentiform nuclei, the deep brain, high energy nuclei, and elevated lactate and pyruvate levels with a normal ratio. The second case is also a female. This is a preemie, now at day of life 28, with dysgenesis of the corpus callosum, a VSD, a cleft palate, and elevated blood lactate. And a third case is a male, full-term newborn, generalized hypotonia, and a brain MRI showing thinning and hyperintensities of white matter, elevated blood lactate and pyruvate, and a normal ratio. This is a PDC talk, so a functional enzyme testing was performed on either lymphocyte or fibroblast, or both in this case. And because of the known, and we've published on this, sensitivity differences between cells and tissue type, you would expect to see some variability in assay results depending on the cell types one uses. And so fibroblast was low in case 1 and 2, but normal in case 3, and low in lymphocytes in case 3. So one would think that this is PDC deficiency, and one would use a ketogenic diet probably as a starting therapy, but my recommendation, at least from our understanding of the variability of this disease, is a molecular confirmation before starting any sort of ketogenic diet on any patient with PDC deficiency. So further testing with KDC on fibroblasts was not done for 1, but was low on 2 and was normal in case 3. E3 was normal in all of them. So this is not an E3 deficiency, but the fact that KDC is low, this was the complex, the dehydrogenase 6 o'clock of the TCA cycle, is a red flag that says this is much different than the typical PDC due to PDHA1, but something different. So ketogenic diet was started in all 3 in some way or another. The first one lived. The case 2 and 3 died shortly thereafter after starting ketogenic diet. The genetic etiology were different. The first case is typical PDHA1 with a missense mutation. Second case was a lipomide synthesis deficiency, LIAS gene defect. And the third one was a short-chain enyl-CoA hydratase deficiency. It's a valine metabolism / fatty acid disorder. And so we've subtyped these PDCs into 3 different subgroups: primary specific, primary generalized, and secondary. The complex itself is a big complex. It has multiple subunits, E1, E2, E3. E3 is shared by other dehydrogenases. E1 and DLAT by E2 is unique to PDC, as is the PDH kinases and the PDH phosphatases. The E1 is coded for by PDHA1, which is X-linked, and PDHB. And essentially, TPP is a cofactor, thymine pyrophosphate. It binds in the active site. It needs to be activated. And the next step is a decarboxylation step in the active site where CO2 is released. And the acyl group is transferred to E1 on a lipoyl group. There's a modification on the E2 with a lipoyl group. So this lipoyl group is also shared by other dehydrogenases. So anything that disrupts the synthesis and incorporation of the lipoyl group on a dehydrogenase will also have much more of a generalized effect. And then the final step is a transfer to a CoA and release as acetyl-CoA. And then the final step is then reoxidation of the loop-O group to form the disulfide bond. And that is done by FAD, FADH, and then NAD reduction to NADH. So as I alluded to, loop-O group is also found in alpha-ketoglutarate dehydrogenase, those red arrows, oxoadipic dehydrogenase, which is the enzyme complex right proximal to the glutyl-CoA dehydrogenase, which is responsible for GA1. The branched-chain keto acid dehydrogenase also has that, which results in maple syrup urine disease, and glycine cleavage system, which results in non-ketotic hyperglycemia. The H protein has the lipoyl group. And DLD is shared also by these various dehydrogenases in different forms. And their affected pathways is shown on the right. So we subclassify the patients into different groups so that we can tailor the therapies in the future for these different subgroups. And so primary specific PDCD is very much complex protein specific. And the highlight of that is the PDHA1. The second generalized is, you saw an example with an LIAS. The secondary one is this short chain enol-CoA hydratase. And I circled this in red for TAS, which is tafazin gene, which causes Barth syndrome. And because we identified with the newborn screen a patient, a newborn with satisfying the criteria. And tafazin is an interesting gene product. It helps tether the complex into the inner mitochondrial matrix, inner mitochondrial membrane from a research group in Michigan. And mutations in TAS gene also cause functional PDC deficiency. So the structure of the E1 complex, which is one of the complexes of the PDC, is a dimer of heterodimers. So alpha, beta, alpha prime, beta prime. Alpha is coded for by PDHA1, beta is PDHB. And there's two symmetric active sites that is constituted by loop A and loop B. And the thymine pyrophosphate is bound there. And so there are structures that are important in this, what is known as the flip-flop mechanism of action of the structure, where by the time one thymine pyrophosphate is being activated and the carboxylation is happening and the acyl group is being transferred, the other active site is going through the same process. The molecular genetics, again, PDC is a mitochondrial matrix complex, although there are data that suggest that there is a translocation of the complex into the nucleus, where it is involved in acetylation of histones and regulation of genes. But that is how it does that is not clear yet. There is more than 38 genes associated with the functional PDC deficiency, but almost up to 88% of cases are due to mutations in this X-linked PDHA1 gene. About 5% are due to other primary specific genes, PDHB, DLAD, PDP1, and the PDHX. And the rest of the 5% are a mix between much more primary, generalized, and secondary conditions. Of the cases with PDHA1 mutations, about 60% of them are due to missense mutations. And arginine residue replacements constitute between 40 and 50% of all missense cases. And there are recurrent replacements. And these arginines 234, 273, and 349 constitute about 57 to 80% of all arginine replacement. And specifically, this arginine 349 constitutes about a quarter of all arginine replacements and about 10% of all missense mutations in PDHA1. There is gender predominance depending on which arginine is replaced. And as you will see in later slides, there is an allosteric impact of replacements on the active site of the complex. So the active site of the loop A and loop B are altered by replacements that's happening further up in the further away from the active site. And I'll talk about these next two lines where up to 88% of all the disease causing missense mutations are solvent inaccessible, i.e. buried, and that 70 to 75% of those buried missense mutations are in a specific region of the protein and noted here, and namely between exons 5 and 7. So Nikki Dusich did this work actually, and she was interested to know what is the solvent accessibility of those amino acids whose replacements causes disease, missense replacements cause disease. And she found out that about 86% of all disease-causing replacements are buried within the E1 complex to different extents, as opposed to neutral variants, benign or likely benign ones. And then she focused on those that are buried ones. And by comparing the SASA measurements from the E1 heterotetramer versus E1 alpha monomer, one can see whether a specific replacement is buried in an E1 heterotetramer as opposed to E1 monomer. We were able to identify 30% of those replacements that are now designated as subunit interface contact specific residues. And so that is of importance and significance. And in a different independent way, she did a nearest neighbor analysis where she walked across the amino acid from the N-terminus and then asked the question, what are the nearest less than 5 angstrom amino acids to this alpha subunit? Which beta subunit or which alpha subunit, alpha prime subunit, or beta prime subunit amino acid is nearest to as she walks across the E1 alpha. And these blue horizontal line describe those specific amino acids that are closest, and the red ones that are closest to the alpha. And the other blue line, horizontal line, is the beta 1 that is closest to the E1 alpha. And as you notice, all of those disease-causing missense mutations that are involved with subunit interface contacts are also fall within those nearest neighbor analysis. And most of them are centered around this residues R122 to 224. Because disease-causing missense mutations and E1 beta, PDHA and PDHB mutations are rare, we were able to map where the nearest neighbor and which ones are expected to be close together in terms of nearest neighbor. And we were able to identify some missense mutations that are also involved in subunit interface contacts. And lo and behold, we were able to find individuals that have, let's say, a tyrosine 132 mutation in E1 alpha and a methionine 71 in E1 beta. And another pair was between different alpha subunits, between asparagine 171 in alpha and glutamate 177 in the alpha 1. And we asked the question, with these patients now, mutations in different nearest neighbor amino acids have similar clinical presentations? And indeed they do. And this is for the methionine 71, tyrosine 132 in different subunits. They both had neonatal lactic acidosis, lead disease, seizure, and the other pair had, again, neonatal lactic acidosis, lead disease, and severe psychomotor retardation. So the hypothesis holds. Ketogenic therapeutic options. Ketogenic diet is the main therapy, again, for primary specific PDC deficiency. It may not be effective or useful and maybe even harmful if used for secondary conditions or more primary generalized PDC deficiency. It has a positive effect in terms of epilepsy, ataxia, sleep disturbance, speech and language development, social functioning, and reduces the disease burden in terms of frequency of hospitalization and longevity. Efficacy, though, depends on the burst severity and early intervention with KD, and attainment and maintenance of ketosis, which can be difficult because of tolerance. And the ketogenic diet is an untargeted diet or therapy. Dichloroacetate is also untargeted, but it's used mainly for primary specific PDCD and phosphate deficient one, and it's in phase 3 trial with more than 30 subjects enrolled, and it's going to be closing soon. There's very few cases with timing responsive subjects, and other therapeutic options are under investigation with using phenylbutyrate. We have a study where the trial is pending for using triheptanoin for primary specific PDCD, and our work with target-based, and this is the triheptanone is also in Pittsburgh here that will be started, and target-based small molecule options under investigation, and we'll talk about that more. I just recently heard of a gene therapy of a group, a European group, that are trying to try it on 11-month-old with PDCD, but I don't know the details. So good segue into newborn screening. So Anisha Verma is a medical student who is tasked with looking at over 11,000 cases in UPMC that have had amino acid analysis completed, and looking at where that diagnosis is for these patients and all the clinical information, and over an 11-year span in UPMC. And over this 11-year span, there were 12 patients that were UPMC patients that were diagnosed and confirmed with PDC deficiency. What stands out is their first amino acid that was used in the process of the clinical workup was a median age of about 9 months. Molecular and or functional diagnosis was completed by 14 months of age, and a ketogenic diet was started about 11 and a half. So it shows that we need to push this first amino acid testing much earlier with the biomarkers we have, so that we can identify these kids early and benefit them with the therapeutics that we have in hand and novel ones that are coming. And thus the premise behind newborn screening: early diagnosis, early intervention with ketogenic diet to improve developmental and cognitive outcome, to improve quality of life and decrease the disease burden with less seizures and less hospitalization, and to increase longevity. So obviously, what are the biomarkers to use? Alanine and proline, because they can be elevated. And I was surprised that there are none of the newborn screen programs actually analyze and release the alanine and proline concentration in the dry blood spots that are collected from newborns around the nation. And so I worked hard with the Ohio Department of Health and Rosemary Hodge when she was the director of the lab at the time. She's currently at the CDC, to change the algorithm so that we can quantitatively release alanine and proline information from those dry blood spots. And as I understand, Ohio is the only state since May of 2018 that on a supplemental metabolite page releases that alanine proline information. The other two markers that are of use is leucine and lysine. They are strictly ketogenic amino acids, meaning that they bypass BDC altogether and enter at the TCA cycle at the acetyl-CoA level. So they're not influenced by lactate, pyruvate, alanine, or proline, and they can be used as a normalizing amino acid moving forward. We looked at what are biomarkers that are most sensitive for PDC deficiency, and we had data from natural history study that we had started in Cleveland at University Hospitals. Now it's continuing at the University of Pittsburgh in Pittsburgh, where we had 20 patients with known PDHA1 mutations and 2 patients with PDHB, and we'll look at their amino acid values, specifically alanine to leucine ratio, alanine to lysine, prior to them being on a ketogenic diet versus controls. And we realized that alanine to leucine is the most sensitive biomarker to use. We also found that the combination of proline to leucine and alanine to leucine ratios are even more sensitive to use. And so we put together combined ratio criteria as a starting cutoff of alanine to leucine greater than 4 and proline to leucine greater than 3 to move forward with our newborn screening protocol. And so we had the alanine and proline release data from dry blood spots since May 4th, 2018 in Ohio. We requested de-identified dry blood spot data that shows gender, gestational age, birth weight, NICU stay, TPN, packed red blood cell use, age at time of specimen collection, and alanine, proline, and leucine concentrations only. And then we distributed IRB-approved flyers that was sent to various medical centers, genetics groups, neurology groups, where we advertised the study, and for them to then potential patients with a confirmed PDC diagnosis or other confirmed diagnosis of disorders on the newborn screen panel, mainly organic acidemias or others, or even mitochondrial conditions, that these subjects can contact our research coordinators for screening and eligibility and then consenting. There was no need for any clinical visits, no need for new laboratory blood draw or testing. They were only allowing us to review existing medical records and access their newborn screen test result so that we can associate a name and a specific clinical picture and a diagnosis with these dots that we have de-identified from the Ohio Newborn Screen Lab. And so we got over 123,414 exact, actually non-duplicate and data complete dry blood specimen information. So complete in terms of all of those parameters that we requested, because it was important to make sure that TPN is not being used, or this kid did not get packed red blood spell, or that because those will alter the actual leucine levels, because it's leucine and lysine are essential amino acids. And so we looked at greater than 99.9 percentile for alanine to leucine or proline to leucine. These are red spots. And so there were only 217 of them. And if we add the criteria that we talked about to these parameters, then we're down to 145 green spots. And the number of term infants and the number of infants in NICU in these two populations are the same. And within this group, then, we found 3 PDHA1 patients and 2 patients with primary mitochondrial conditions. We extended that study a little longer, and then we were able to find 1 TAS mutation patient in there and 1 patient with a combination of a nuclear mitochondrial mutation and a mitochondrial DNA mutation that presented itself as a mitochondrial condition, and a kid with sepsis. And so the positive predictive value we calculated was at least 2.8%, and that is comparable to what we see for Krabbe disease in Ohio, much better than that. Ohio's Krabbe disease PPV is 1%, Hurler is 3.3%, and so it's comparable to Hurler. And the Pompe disease has the best PPV of 18.5%. So even though it is sensitive but not specific, we anticipate that adding a second tier of targeted molecular testing that targets primary specific themes, PDH1, PDHB, DLAT, PDHX, and PDP1, would then identify those patients that specifically have PDCD due to primary specific gene defects that can benefit for early intervention of ketogenic diet and other novel targeted therapies, as opposed to identifying other kids with mitochondrial DNA, primary mitochondrial conditions for which we don't have therapies as of yet. So these were the details of the patients. Two of the 3 patients with PDC deficiency were started early with ketogenic diet and are still living. One of them is my patient that is also enrolled in DCA trial now. The 3rd patient with PDCD came in metabolically decompensated at the hospital at 3 months, and parents elected to redirect care, and thus ketogenic diet was not initiated, and she passed away. Switching to target-based small molecule therapies. So both Nikki and Hattie, Nikki was involved in identifying, both by the subunit interface contact amino acids, and these red spheres are those that are due to E1 alpha replacements that cause disease, and the blue ones are PDHB or E1 beta replacements that cause disease. And the 2 of interest to us that are both involved in SSIC are the R349 and tryptophan 185. And this is where the loop, the active site is behind this structure here, and the other active site is in here in the alpha prime. And so you can see that they're at least 15 angstrom or more away from the active site, yet they're exerting their effect on the structure and function and causing disease. So we wanted to understand how it's doing that. And Hattie, a postdoc working with Oles Isayev, my collaborator at the Mellon Institute at Carnegie Mellon, is a computational chemist, and she said we can do this with looking at backbone C alpha fluctuations and whether there is any mobility changes anywhere. And we've done this with replacements of both the 349 with histidine or cysteine on both alpha subunits. Therefore, we're mimicking a little bit of what the genotype of a male patient with an X-linked disease would be, where both subunits are replaced. And we've also done that with only replacing one subunit, the reflecting female phenotype, and we see similar things. And the fluctuation and mobility of the loop A is affected. It's either more rigid or more flexible. And this sort of phenomenon was also observed when they did specific phosphorylation of specific serines. There's two important serines in loop A and one in loop B. When you phosphorylate, it causes a rigidity of the loop A and thus functional deficiency in the complex. And so we were able to replicate this for mutations that were far away from the loop A active site. She also did principal component analysis and cluster analysis that looks at the large-scale motions that are usually the most biologically relevant. And you can see in the wild type here that, principal component 1 and 2, major components of motion, they form a kind of a horseshoe kind of a figure with an open loop alpha prime structure and a closed loop alpha prime as shown here. And this is the loop of the alpha prime that is moving, and the other one is right behind in the pink area here. And then when you replace both amino acids, both subunits with the cysteine is at the position 349, you can see the shift is magnified in this alpha prime, and it's shifted completely in the alpha. And then the picture is also a V shape instead of the horseshoe shape. And so this measures both the magnitude and the direction of these shifts, where in the alpha loop A region, the mutation is orthogonal to the wild type direction. But in the alpha prime loop A, it's the same direction, but it's magnified. The magnitude is higher. So this is the same. Because of time, I'm going to limit this, but we've seen the same sort of a behavior when we replace 349 with histidine, either at one subunit or both subunit. And then what we wanted to do is to look at how the communication network is altered. These are interactions between residues between subunits. And we were able to measure that using something called eigenvector centrality analysis to look at whether these communication networks are being altered by disease-causing missense mutations, and indeed they were. And these same regions that were showing changes were indeed the same regions that Nikki had found by nearest neighbor analysis in SASA, where these replacements in there cause disease. So we have shown that missense replacements greater than even 15 angstroms away from the active site do trigger subunit interface contact changes, which lead to known reduced PDC activity in disease. So Hati was able to then look at the various conformers, and we were hypothesizing, and Nikki did, with that an arginine 349 replacement is going to generate a pocket because of the way these are superimposed histidine 349 and cysteine 349 changes. And this pocket generates a very definable measured volume that is amenable to small molecules that can dock there and bind there. And the question is, can we have and find a small molecule that restores the missing interactions between the subunit interfaces that then allosterically impacts the active site and restores PDC function, and thus generating a target-based specific small molecule that can have therapeutic implications. And this small molecule, of course, has to go across the blood-brain barrier. We can look for those specific molecules and also go into the mitochondria, which we can tag them to go in there. And so we're approaching this in two ways: by the traditional virtual screening of greater than 100,000 CNS-focused library, and also by machine learning, deep learning algorithms, where the machine will design a new drug that targets that 349 with the favorable parameters of blood-brain barrier properties easy to synthesize, and optimize binding, and see which one is better. And then we can compare and optimize the R groups accordingly. Then we have a method of doing molecular dynamic simulations, and we have the data already. We want to see which one of the targeted molecule then restores this perturbations that we're seeing down to wild type level, both in the fluctuation and in the principal component analysis or the centrality analysis. Then we can see if we have that molecule synthesized already, or we can synthesize it. And then we can test that in vitro on cell extracts from fibroblasts or isolated mitochondria from fibroblasts with known and well-defined PDC activity assays, and also looking at restoration of the redox balance by measuring NADH and NAD ratios. And this is ongoing right now. My research helps to advance newborn screening research, and also the aims and mission of the Newborn Screening Translation and Research Network, and by identifying biomarkers for PDCD newborn screening, and by piloting PDCD newborn screen for potential future inclusion of this disorder in RUSP, and also in discovering novel untargeted and target-based therapeutics for PDCD. And with this, I'll end with my acknowledgement slide. And there is a lot of people to acknowledge. I'd like to acknowledge most and foremost is Doug Kerr, who spent most of his career studying PDC deficiency, and who has been my mentor and my colleague who picked my interest in PDC deficiency. I've been involved in that now for greater than 10 years. And collaborators, Jason Mears in pharmacology at Case Western Reserve University, who worked very closely with Nicole on the molecular modeling, and my new collaborator, Olis Isaev from the Mellon Institute at Carnegie Mellon, who with his postdoc and the graduate student, Paulina, who is actually involved more on the docking and screening, are very much interested, and I thank them for being open to studying a dehydrogenase, because his expertise is in studying kinases, and it's easy to inhibit a kinase. But the question is, we want to restore the function as the dehydrogenase with a different concept altogether. And so I'd like to thank the Newborn Screening Translation and Research Network for inviting me to be part of their group. I'm going to enjoy being part of their group and seeing how my research interests mold and integrate into the whole network, and hopefully there will be interest in PDC deficiency and newborn screening by my involvement with the group, and which will translate into better care for these kiddos, and decrease PDC burden, disease burden, and longevity. And part of my motivation actually in target-based conditions, therapies, is because at what we've seen with target-based therapies, small molecule for cystic fibrosis, where the CFTR monomer protein gating function was restored by the Paragon molecule evacaftor, that is targeted specifically for the G551D mutation that only makes up about 2 to 3% of cases with cystic fibrosis. Yet that has been markedly improved, the disease burden and longevity and quality of life on these patients with cystic fibrosis. And FDA just recently approved a factor, or at least a combination of 3 small molecules in the same tube, that now covers 90% of gene mutations in cystic fibrosis, including the most common delta F508. So I'm hoping that by starting with the R349, a targeted small molecule, we can then add other small molecules into the mix and be able to benefit more than 10% of patients with PDC deficiency and reach almost 50% if we can. Thank you for your attention. I'm open to questions now.